This is lecture 10 in Principles of Metabolism, and in this lecture we will look at respiration on the cell level. So what is respiration good for? Well, we can start by looking at NADH. So we have seen in the TCA cycle lecture and in other lectures that a lot of NADH is produced when we oxidize substrates. That's a common way of capturing energy. And NADH in itself is a highly energy-rich molecule. So if you would go on and oxidize NADH in turn using oxygen as the electron acceptor, that releases a large amount of energy. The delta G of this reaction, oxidizing NADH in the presence of oxygen, releases about 220 kilojoules. So that's a lot. That's several ATP. So how should we do this? Is there some enzyme that can couple this to the production of lots of ATP? Well, it turns out there is not. This is a large amount of energy, and nature has somehow chosen to split up this oxidation into several steps. So we will look at those, and these form the basis of the respiratory chain. Now, when you look at the respiratory chain and these type of oxidation reduction reactions, you often encounter these type of voltage differences. So instead of the delta G, you have a delta E, so an electric potential difference, which is measured in volts. This is pretty much another way of measuring the same thing. You're measuring how the energy changes when you go from a more reduced metabolite to a more oxidized one. And this is because we're talking about electron transfer reactions, so you can measure this in volts actually. So it's just a different way of measuring it. We're not going to deal with it much, but I just want to mention it because you will probably see it. And you can convert between the delta G and these delta E's in this way, counting the number of electrons and using a constant known as Faraday's constant. But anyway, this is a highly energetic reaction, we said. And so what nature does is that it uses a sequence, a chain of redox reactions, which ends with oxygen as the terminal electron acceptor. So oxygen is where all the electrons go in the end. And it looks like this. So NADH first reacts with a quinone and reduces this, passes the electrons onto that, so you get a quinone with a couple of hydrogens on it. And this has still a fairly large delta G, but more manageable. And then in the second step, the quinone reacts with a cytochrome C. This goes from an oxidized state to a reduced state. And again, you get a few kilojoules out of this, 32 kilojoules per mole from that. And then in the final reaction, the cytochrome reacts with oxygen. It gets oxidized, and the oxygen actually becomes water. And this is still a fairly energetically sort of rich step. So the delta G is large here. You get 150 kilojoules per mole from that. But this is the sequence that we find in biochemistry. This is how it's split up. So now we have several sort of intermediate steps in which we can extract energy from NADH. So then the question is, how do we capture this energy? Can we link this directly to ATP production now from these reactions? Well, for a long time it was thought that this was kind of how it worked. There might have been some other intermediate, we thought, but probably it's something like glycolysis, right? You're going to make ATP from these reactions. But the reality turned out to be a bit more complicated. So what actually goes on is a process called chemiosmosis. So the energy that we extract in these oxidation reactions are converted into potential energy. So there are these respiratory complexes that take care of these redox reactions, passing the electrons onto oxygen and finally making water. And these reactions simultaneously store or capture the energy by pumping protons. And these end up in the intermembrane space of the mitochondria. And so this is a potential energy form. You build up a sort of pressure of protons, and this is used by ATP synthase. So this pressure of protons can kind of act by using ATP synthase as a kind of turbine to extract the energy and drive ATP synthesis that way. So this is a phenomenally complicated system when you look at it, and if you would propose this, people probably wouldn't believe you. And that's exactly what happened when Peter Mitchell proposed this. Nobody really believed him. But the biochemical evidence in the end was overwhelming. This is actually how it works. So very interesting system. So are there any advantages of having respiration or the oxidation of NADH set up this way? Well, one advantage is that this acts as a kind of hub. There are a lot of different substrates or sources of energy that you can feed into this system. We looked at this bow tie model of metabolism, and this is probably really at the heart. This is the most central process you can think of. So we say that NADH can enter this system, and it can follow this path and pump protons. But at the step of this oxidoreductase, this redox reaction here, where the ubiquinone, the Q, is accepted, here there is also a lot of other complexes actually that can enter. So you can see that there is energy that can come from various parts of metabolism that can enter here. And everything in the end ends up donating the energy to this proton gradient, building up this form of potential energy, and you can make ATP out of that. So it's kind of a way of collecting a lot of diverse processes into one place, kind of hub. Fatty acids is probably one of the most quantitatively important of these other processes. So the fatty acid oxidation works via FAD, and this works through another redox enzyme called the ETF dehydrogenase, and this goes in this way. A couple of these other processes are probably minor. There's a dehydrogenase for glycerol. There's one that is active in pyrimidine synthesis, and I'm sure there are a couple of others which I haven't included here. But overall, we can see that this is a way of organizing metabolism, oxidative metabolism of various substrates around this hub of proton pressure as a form of energy storage. So we all know that the respiratory chain and respiration occurs in mitochondria. But I think the textbooks are still a bit unclear as to exactly how the structure of respiration, how it really looks. So there are many places where you can find pictures that look like most of the respiration are sort of on this part of the inner membrane of the mitochondria, and they put protons in the intermembrane space, and then the ATP synthase is here somewhere. That doesn't make that much sense really, because the outer membrane is full of these pores. So if you would try to build up a proton pressure here, it would probably leak out pretty soon through the outer membrane. So some of the drawings that you see of the respiratory chain, I think, are a bit misleading. So what we are finding when we look at structure studies where you look at the ultrastructure of mitochondria and where you actually find the complexes, it's that the complexes in the respiratory chain tends to be in the cristae of the mitochondria. So these are these classic mitochondrial pattern, these membrane parts that are on the inside. And if you zoom them up, they look kind of like this. They form a closed space. Maybe a bit of leakage here, but it's pretty tightly closed, where you can have a closed space where you can build up protons. And this is where you actually find the respiratory complexes. So that makes sense. So the respiration system is really kind of it's a closed system. These complexes that pump protons pump it into a closed space, and therefore you can build up this pressure, and you can use that to synthesize ATP that way. Now there are probably some complexes also that are on this side of the inner membrane, and there is some pH difference across this membrane, and that is used to import molecules via solid carriers and things like that. But overall, this is the structure that seems to be the right one. So when you look at the stoichiometry of respiration, this gets a little interesting, because if we think about regular reactions like ATP synthesis in glycolysis, you always have a fixed stoichiometry. You can put numbers on this. You make one ATP out of the reaction that consumes one phosphoenol pyruvate and so on. So you can put fixed numbers on this. When it comes to respiration, it's a bit more slippery because you have these pumping of protons, and then you have this physical phenomenon of a proton pressure, and then it goes through this channel, and then you make ATP. But you don't have really clear numbers on stoichiometry here. So then this is a source of the debate of how much ATP you actually get out of respiration, that these actual numbers are not straightforward to estimate. So if you look at it, the part where you go from NADH down to oxygen, so the actual sequence of the redox reaction, that is fixed because that's just electron transfer. That's a sequence of reactions, and you can write down a formula for that. So you have a fixed stoichiometry, so one NADH will reduce half an oxygen because there's a pair of electrons reduces half a molecular oxygen molecule. So that's fixed. But the other part, to actually make ATP, that is variable because that depends on the proton pumping, and this is a sort of more messy physical process. So that's variable. But we think that in most cases, this ratio is about 1/2 of an oxygen, so you can say one oxygen atom to 2.5 ATP. This is sometimes called the PO ratio, so the ratio of phosphorylation, meaning synthesis of ATP, to oxygen, the PO ratio. This is about 2.5. That's from NADH. If your electrons are entering from the quinone, so that's a bit later in the sequence and you don't pump as much protons in that case, so then you get about 1.5 ATP. So it's a bit small, a bit less. And you can use these numbers then to figure out roughly how much ATP you get from processes like the TCA cycle. So for example, in pyruvate oxidation, if you make 4 NADH, well, if you do the numbers here, that should account for 10 ATPs. And then you have one of the quinones, so that's 11.5, and so on. Yeah, these numbers can differ a bit depending on where you look in the literature because the stoichiometry is variable. Now this thing with a variable stoichiometry is kind of interesting because it means that the respiratory chain kind of has the ability to adjust how much ATP you get out of your substrates. So why could that be useful? Well, respiration is not only a source of ATP, it's also known to be the source of most of the metabolic heat production that we have in mitochondria and in our bodies. So again, this review we mentioned before by Rolf and Brown, Physiological Reviews, goes through this in quite some detail. So here we are representing the respiratory chain, ATP synthesis and usage in a single sort of compact figure. So you have whatever substrate you're oxidizing, you're making NADH out of NAD, that would be the TCA cycle. This is the redox reactions in the respiratory chain. You're pumping protons. The protons sort of fall back, and that helps you synthesize ATP. And then there is a whole bunch of reactions that can use ATP for all sorts of work that the cell needs to do. And then ATP comes back. This is kind of a very brief summary of all of cellular metabolism. So if you ask where the heat comes from in all of these reactions, I mean, all of these reactions are not 100% efficient, so there's heat loss everywhere. But where is most of the heat loss? Well, people are trying to figure this out and do various experiments and measurements and calculations. And what we come up with is that most of the heat loss, the majority, is in this part. The respiratory chain, this section, the proton leak through the membranes. There is always some protons that slip by the ATP synthase. And the ATP synthesis itself in the ATP synthase in the mitochondria. So there's a lot of heat that comes from here. And then a portion of the energy is captured as ATP, of course, and then that is used. And because this is a substantial source of both ATP and heat, being able to adjust the relative rate of each is kind of interesting. And the adjustment physically is done by the mitochondria can regulate a bit how much leakage you have of protons that sort of comes back into the matrix without synthesizing ATP, which means that the mitochondria are able to some extent, adjust the yield of ATP. And that we talked about before, this energetic phenomenon that the yield of ATP also affects the rate at which the process goes. So this is a trade-off, yield versus rate. And so mitochondria can actually adjust this trade-off, whereas a process like glycolysis, which has a fixed stoichiometry, cannot do that. So we don't know, but this might be an advantage to the usage of respiration. And heat production in itself is, of course, also beneficial in many cases. Cells and animals need to keep warm. So heat is not purely a waste either. So these are some features of energy production in the mitochondria.